Good morning, one and all present here, respected principal, distinguished speaker, esteemed faculty member, researchers, and dear students and participants. I feel honored and privileged to welcome you all to this two-day national conference on ethical and scientific consideration in animal research, organized by Department of Pharmacology and Internal Quality Assurance Cells of Institute of Pharmaceutical Education and Research, Borgaon Mege, Varda, and sponsored by Committee for Control and Supervision of Experiment on Animal, Delhi. Animal experimentation has significantly contributed to the advancement in pharmacology, toxicology, biotechnology, and clinical medicines. Many life-saving drugs that we are using today are the result of carefully conducted animal studies. In India, research involving animal is governed by the guidelines of Committee for Control and Supervision of Experiment on Animal, that is CCSEA. At the institutional level, the Institutional Animal Ethics Committee, that is IAEC, play a crucial role in ensuring that research protocol comply with the ethical standard. This conference has been organized with the objective of promoting awareness of the ethical guidelines, strengthening scientific validity in the experimental design, encouraging human research practices, and discussing emerging alternative to the animal testing. As academicians and researchers, it is our collective responsibility to afford both scientific excellence and ethical accountability. I am confident that today's deliberations will provide valuable insight, strengthen our ethical commitment, and guide us toward more responsible and scientifically sound, richer practices. Let us use this platform not only to exchange knowledge, but also to confirm our dedication, consideration, integrity, and innovation in research. With this positive spirit, now we move towards the official inauguration of this conference. I request Dr. R.O. Ganjiwale sir to inaugurate the conference and express his views.
Thank you.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम्.
Now I request Dr. R.O. Ganjiwale sir to express his views on for this conference.
Thank you, Dr. Avari. Good morning, one and all, the respected speakers, faculty members, and the students. Indeed, it gives me a great pleasure to welcome all of you to today's international conference on ethical and scientific consideration on the animal research. And for our renowned speakers, as all speakers of national and international reports. I welcome Dr. R.K. Goyal, sir. He is going to talk on the medical experiments on animal without compromising on outcome and human consideration, advances against the challenge. Thank you, Dr. R.K. Goyal, sir. Second speaker, Dr. Samiksha Saklami. She is going to talk on role and the functions of CCSEA. Then Dr. Richa Rautela. She is going to talk on regulatory framework for animal experimentation in India. Dr. Papia Bigonia. She is going to talk on the ethical use of animal in biomedical research, practical implementation of 3R concepts. And Dr. Prahlad Wangikar, he is going to talk on ethical use of animal in research and concept of new approach methodology. On behalf of the organizing committee, I heartily welcome all the renowned speakers. Dear friends, as you know, we, this animal research has really played a very important and a vital role as far as the development of the medicine and the various pharmaceutical product it is concerned. Without clinical study or without animal research, we cannot successfully develop any particular pharmaceutical product. And the theme of the today's conference, it is based on the two parts. One, that is the part of ethical consideration, and second, it is the part of the scientific consideration that we have to keep into mind while performing the animal research, particularly in the pharmacology lab, whenever we do the pharmacological screening of the various crude drugs. So far as the ethical consideration of this animal research it is concerned, here we have to follow the rules of the 3R. One, that is no replacement. Second, it is the reduction. And next one, it is the refinement. If you follow very seriously this 3R principle, then to this ethical consideration will be observed in the while performing the animal research. Because as far as the part of the ethical research it is concerned, here we have to take into consideration the animal welfare also. Because while performing the any experiments on the pharmacology, it should not cause much more pain or much more harm to that of the animal. As far as possible, in the reduction part, we have to see that the minimum number of the animals will be utilized while performing the experiments. After this, again, there are so many other parts, scientific considerations are there, because whatever the international and the national experimental protocols are we given, those experimental protocols we have to follow. Without taking your much more time, I again express my very sincere thanks to all the speakers and my special thanks to the CCSEA for considering the proposal for the sanction of these institutes. And with these words, I wish a conference a grand success with the fruitful deliberation. Thank you very much.
Thank you, sir, for your valuable and insightful talk. Without taking much of your time, now move forward to the most awaited session of today. It is my privilege to introduce our keynote speaker, Professor Dr. Ramesh K. Goel. Professor Ramesh K. Goel, Professor Emeritus, Institute of Human Genetics, was the former Vice Chancellor of Maharaja Sayajirao University of Baroda and Delhi Institute of Pharmaceutical Science and Research. He is an honorary professor at State Medical University, Russia. He has 6 patents, author 48 books and book chapter, and over 450 full research articles. H-index is 55. He has guided 51 PhD and over 220 postgraduate students and recipient of 80 awards for research contributions. Currently, Dr. Goel is president of International Academy of Cardiovascular Science, India section. He is the president of Indian Pharmacological Society, Society of Pharmacovigilance India, and Indian Society of Hypertension. He was instrumental in development of formulation for COVID-19, duly approved by Ayush Government of India. Professor Goel has 48 years of experience in teaching and research. He was a postdoctoral scholar, visiting scientist and visiting professor in University of British Columbia, Vancouver, and University of Canada. He has been listed amongst career-based top 2% scientists of the world in the field of pharmacy and pharmacology, subfield cardiovascular science, as per the list published by Stanford University. He served as the expert member of Indian Medicinal Plant Review and member of Scientific Advisory Committee, New Delhi, and Pharmacovigilance Center of Indian Pharmacopoeia Commissions, Faridabad, and the phytochemical groups of Central Drug Standard Control Organization, that is CDSCO, New Delhi. National Board of Accreditations, NBA, and peer members of chairmen in NAC committee. He is the chairman of Scientific Advisory Group of National Dope Testing Laboratory of Sport Authority of India. He was the chairman of Central Region of All India Council of Technical Education, that is AICT, and also the executive member of AICT. With this brief introduction, I request Professor Dr. Goel sir to proceed with the sessions.
Thank you, Dr. Ahori, for giving the introduction. But I would like to please allow me to share the screen.
Yes, sir.
Sir, you can share it now.
Okay. Are slides moving?
Yes, sir.
Slides are moving also, no?
Yes, sir. Yes, sir.
Okay. Once again, thank you very much for giving me this chance to speak on the animal welfare in particular. And there has been talks all the time that why all these things are there, should it be there or not? So many times these questions have been there all the time. And I have spent almost 48 years in research, pharmacology research. I remember those days also when we used to use animals freely. And now with the regulation, when the regulations started in 1990s, 98 to be very precise, we were also opposing, like, no, what is this? And somehow what I learned, what I felt, what have been my experiences, how I take these regulations, I would like to present this in today's lecture. Well, it's not moving. Okay. So use of animals, when you think of pharmacology and animals, it has to go together. I mean, it has been going from ages. The pharmacology subject started with the development of a lab by the first pharmacologist, Rudolf Wachem, in his own house. He was doing the experiments to prove that, yes, how curare affects, how various plants they produce the effect. That was the beginning of the whole part. So animal experiments have been going on from ages. Well, in pursuit that, if I say that, like, if we go really in the very much of history, Aristotle, I mean, before first BC, he used to dissect animals. That was for anatomy, learning anatomy. Then demonstrations on pigs, vivisection. It was very well known. Then the experiment which was done for the blood pressure and the movement, how the blood occurs, how much is the blood. It was the William Harvey. They also utilized the same. And this thing has been continued till that. Like, what I'm trying to say is that, yes, use of animals is definitely important. It provides measures for health and welfare. The solutions to many questions have come because of the through the animal research. Today, even the polio research and even COVID, during COVID, monkeys it was a very much a challenge to ICMR and Dr. Balram Bhargava in the movie. I hope people have seen that, some of the wonderful movie which we prepared about the COVID, how the vaccine development takes place. He mentioned what hardship he had, but since he could get any monkeys, he could do the research and we could get the vaccine, and it protected the humanity. So likewise, cat, we can go on talking from end to end, there is no end. But somehow I have a feeling, some people felt bad also, that I used to feel that slowly pharmacology, it developed as a branch, as a subject, and it is disappearing also fast. In the sense that the drug research, when it comes, it is more biotechnology based. The whole thing has changed. And all Nobel Prizes are coming to biotechnologies rather than the real hardcore pharmacology. It's going on. But yes, pharmacology has several roles, but with respect to animals, the biomedical research, what we say, they are used. But we all know that regulatory, when you go to CDSCO to get any approval for drug, they will ask, okay, where are these experiments? Where are toxicology experiments? Where are acute toxicity, chronic toxicity, the anti-cancer activity, and so on? They ask, and you need the data. So it is going on. Then quality control, again, there was a time when the new drug was given to check the safety. And if it shows fall in blood pressure in dog, that means it is something hypotensive, histaminergic effect. And if it is blocked, we'll say, okay, yes, it is like that, it is it. And if drug is not producing the effect, it is good. But now you need not to do that. You simply show that in vitro also you can see. Biochemically also real. When I wrote first chapter in my book of pharmacology about the bioassay of drugs, one of the sentences was that we have to have the bioassays will be replaced sooner or later with the chemical assays or simpler methods. And it is a reality. Over 20 years, nobody even talks about the bioassay. Although the syllabus is there, somebody is pro, but hardly they are used. At least the acetylcholine, histamine, and all, they are gone. Yes, it is used for certain things, certain biotech products again. So that is there. And then finally, the training and demonstrations. There also, the simulation experiments like Dr. Ravindran, Dr. C.R. Patil, they have done it. And it is a worldwide now, and it is a worldwide phenomena, or other places also it has come. Well, so if you see both the sides, then you will realize what is the reality when it comes to use of animals. And has it come? I hope it is not disturbing you, this screen. I hope it is not disturbing you. Okay. Or okay. Stop. No. Okay, so what I'm trying to say is that I am now first going to give you the what are the viewpoints. Scientists, I started with the scientist viewpoint only. How animal activists, they are present today in the seminar, how they feel and what should they feel, how philosopher feel, and as a pharmacologist, how I feel. Well, from viewpoint of scientist, it is vital. No scientist will say that no, it is not required. And alternatives, yes, scientists many times feel, we also feel that in vitro or alternative, the use of like the Drosophila and all that, they have some rather large limitations also. But ultimately, you have to come to in vivo also. So you cannot get away from that. If not animals, people say, why to do in animals? Why not to try in human when the Ayurveda has written so much, the different systems of medicines have written? Why you have to do that? Well, but they agree that no, you cannot harm the human being. So you have to start with the animals. But animals also cannot be harmed. That is also a reality. So, I mean, is it possible to minimize the reduction that comes into picture? But yes, they have started agreeing. Everybody agrees now. There is no question. 25 years have passed after implementation of the regulations. And they are the biosystems and they are used. Now, how animal activists feel? Now, animal activists, they feel that they cannot see the sufferings of the animals. Well, I will not go into philosophical that what about the sufferings of humans and all that, but okay. And alternatives must be thought on. And today we have several alternatives. If not alternatives, the modifications of the approach in the drug research. My focus will be in the entire lecture about the drug research, that the new drug discovery, development, how are they impacted, and can we do without compromising, as I mentioned the title, the outcome of the research should be there. That's what is there, right? So activists say that superior human life does not confirm right to exploit the lower order. Yes, correct. We cannot see the animals being killed in an improper way or like that. And the concepts of 3 Rs is well accepted now, but animal activists, they emphasize on that. Why I'm saying is that when it comes to ultimately the CP or CCSEA regulations, now they tell heavily on the about the ethics committee, and they are one of the important part. Philosophical is a sort of a bridge between the two, scientists and the activists. Yeah, we do understand whatever both the sides are saying. It is important animal experiments. At the same time, it is important to see that the sufferings are also less. Now, I am a pharmacologist all throughout my life, 50 years almost. And if I start my PG days. So, as a pharmacologist, as I told you, the whole history, if you see, it started first with the isolation of compounds rather than the real direct testing. But afterwards, like Claude Bernard, Rudolf Buckim, they made their own labs. And the whole idea was at that time to find out how they act. And that's what they used to see in the animals. And it continues. But so two things continued: isolation of compounds, and other is the activity evaluation. Whether it is a pharmacognosy person or a chemist, the path remained the same all throughout. And it is talking about 18th century onwards. So 18th, 19th, and now 20th century also. Now we are in 21st. So in 300 years, so many compounds are available, so many activities are known, and methods to do that activity, they are known. So, although I will tell you little later, why to waste time in just seeing the things again and again the same? Whether it is isolation also, whether it is activity also. We'll see that. Now, the respect to animal, I personally believe yet it has to be there. See, when I was VC in the Delhi, in front of Animal House, the whole building is covered with such posters. We respect that, yes, how important are like, for example, simple mouse. The mouse is important because it is an experienced lifesaver. I've just given, one should have the idea that they have been used for what. And because of mouse, how many Nobel Prizes have come? It is all the sacrifice of mouse or sacrifice of mice at different stages. They have brought Nobel Prizes all throughout in one way or other. Same thing goes for the rabbit. Now, rabbit, a docile bioreactor. You can see the research areas wherein rabbit is used. And do we really have alternatives everywhere? Probably not. But yes, the search is always on. But yes, it is there. And it is a sort of a biography we have written now. And again, Nobel Prizes have come because of the sacrifice of the rabbits. You can see how many things got Nobel Prizes. It is all, if animal experiments were not there, it would not have occurred. Same thing goes true for rat. They are the most common: rat, mouse, the rabbit, and guinea pig. These are the four commonly used animals. And if you really search, you will find that large number of discoveries occurred. The work is still going on, and the Nobel Prizes have also come. So the question is, yes, for teratogenicity, for example, we need rat. But for everything, you need not to do teratogenicity. Through the biotechnology, through the developments, we all know that how, I mean, from in vitro itself, you will know from the simple experiment, simple biotechnology experiment, you will know whether this compound is going to be carcinogenic or not. So why to do that unnecessarily? That is also there. If it is missed out, there are questions will remain, but still, said like bioassay of insulin. Now you need not to do even for demonstration or anything. Bioassay of insulin, yes, we have better methods, better analytical method, because we used to do for analysis, to standardize, for quality assurance of the formulation. So there the thought process are important. And so what I just tried to compile and projecting is that yes, for infectious diseases, yes, guinea pig has been again a very important animal for the research, and again contributor for various Nobel Prizes. They have come because of that research. Even hamsters, although less, but it is less common, but still hamster have also been used, and it has shown that. So a time will come when the alternatives, the zebra face and all, they are going to give something extraordinary. That is for sure. Okay. So now, so having said, if I have to just conclude whatever I have said in one sentence, animal experiments are important. They have contributed a lot to humanity. That is for sure. But now let us think of the welfare. Yes, we have to think of welfare. If I go back again in the history, the first legislation came in 1641. That was for the cruelty to animals, and it was passed by the Court of Massachusetts in US there. Again in 1820, law was passed in England. After that, in other part, there was an association formed and American Vivisection Society formed, and it went on. When we think of Indian scenario, we were not behind. Yes, in 1890, the act to prevent the cruelty to animals was there. Implementation, how it got replaced, how sub-regulations came, it is there. Like CPC-SEA, we used to call for the purpose. Now this word purpose has been removed. So CCSEA came into existence. It was there, but in true sense, it started with the move in the 1900 or around, from 2000 onward for sure. And again, there has been revision in 2023, but the important thing CCSEA has given is registration wherever the animal experiments are to be there. And even the breeding also has to occur after the permission and in a proper way. Animal house facility, just like the human clinical trial, the clinical research center has to be approved. The ethics committee has to be approved. So similar rules have come in the animals. And there are subcommittees for larger animals, smaller animals, and all that they are there. So CPC is there. And again, the amendment, the latest amendment which has come in 23, and that is to streamline the process of inspection. There were many changes even before also. When in 2000, I remember when around 2000, when the CPCA thing started, I remember one meeting in In Delhi, it was with Maneka Gandhiji, Honorable, she was Minister, Honorable Minister at that time. And we pointed out at that time that the nominees which you are keeping, the committee which you are keeping, you are not keeping anybody from pharmacy or pharmacology. They agreed immediately. We told that those who are nominated, they have to know something about the animal experiments. Slowly things have come. The veterinarian also got introduced. So things have come, just not that you are simply shouting that, okay, slogans and all that. That thing has gone. Now there is a definite good cusp. But again, during inspection, what to see, how to see, what should be protocol. So that process has been made more objective, I will say, or something very SOPs have been developed, rather we can say SOP for research protocols for large animals also. Because this thing has been there that smaller animals, for the larger animals, that process were tough. And rather it almost vanished. And the neighboring countries like Nepal and all, they were taking advantage of the large animal experiments. So those things have been slowly coming to change. There is a change in that. Now, when it comes to animal ethics committee, so we also, as I told you, in the beginning, when all these things came, even I was also suspicious. I used to say that, is it really required? Why should we do that? There were problems. And, but the reality is that today, animal experiments, they are strictly monitored. They cannot be done just like that. You have to have all the norms of CCSEA to be followed upon. That is there. And it is going on, and you may be having punishment or you may be having problems if you don't do that. Even the journals don't accept. Even I remember those days, what to do when there is the topics are given in syllabus. So slowly PCI, MCI, all those councils, they slowly and slowly got away from the animal experiments. And they have given alternatives to that. Now, again, alternative and simulation, they are important, no doubt. But you have to see that you cannot compromise with certain learning processes also. So, like I remember, we were myself and Dr. C.R. Patel, we were revising the book of our practical pharmacology. The question came, that now they have simulation experiments. If it is a biochemical test, you really need simulation experiments for that. It's a little certain thing. You have to do biostatistics. So there has to be simulation experiments for that. What is this? That is an exercise that can be done in the classroom. It doesn't require that sort of protocols. But yes, it is written, so you have to follow. So sometimes things become little funny also, I will say, ugly thing also. It becomes like that. Anyway, after me, there are many experts, many good speakers are there who will be deliberating. I will not touch upon as a keynote. Of course, you are supposed to cover most of the things, so I tried to cover everything. And so, composition of ISEC, I think every college, every university, they are having it. And nominee from CPC has from the government has to be there. That is, it is there properly, and now they are well trained also. So that is going on. So what this animal ethics committee has to see? What first thing is where the animals are housed, in what condition, the AC and all that. What about the food given to them? After their use, they are rehabilitated or not, or how they are disposed of. One has to see that. animal house inspections. That's why animal house inspection. It should not smell too much. There should be proper entry and exit processes or entry and exit gates. I remember when I joined as a pharmacologist in LM College of Pharmacy in 78, how shabby the house, animal house was. People will not even approach near to that. And now it's not bad. It is like a something which is a VIP type. Food. We used to give any food, some vegetables to the rabbits who used to simply eat that. And same thing was happening for rat and rat. We used to give only gram, the chickpeas or chana. We used to give only the grams in that. I remember very funny thing. I was doing diabetic experiment. One of the students was doing diabetic experiment. He said, say, sir, diabetic animals are not having any high lipid level. It is rather low. And then on the retrospect, we found that the food which we are giving is wrong. We should have given a pellet food. And then the experiments were good. So the experiments may become misleading if proper food is also not given. Disposal, we used to go to the field, dig out something, put animals there and put more sand on that, and that's how. Now incinerators and all, they have started coming, and then again a proper disposal system. So these are the first thing which are focused and they are generalized. But then when it comes to protocols, do you really need such experiment? That is the question. Can you replace? Can you reduce number? Can you refine the process of using the methodology? How can you avoid pain and sufferings to the animal? And then if surgery has been done and if you are not giving the euthanasia or not killing the animal, what you have to do? So these are these sets of questions which come into the mind. Now, as I told in the beginning, when around 2000, we were also against that. But then slowly and slowly, how we felt, that I will tell now. So what has been the impact of this regulation? First, as I told, the most important, housing, the food which is given, how they are disposed of. So I think everyone sitting or participating in this seminar, in the workshop, will agree that yes, now every place you find proper animal housing, food, and method of disposal are well. They are not shabby at all. If they are shabby, that means they are not fit for the experiment. I myself, I can tell what I used to, how I worked up to 90 or 95, and now how the situation is. Definitely, this thing is, it has happened, is good. The bad thing is that since 1999, 90 or 2000 onwards, there were some pressures. Yeah, the pressure was on PCI, stop this, change the syllabus. And it was done. Or create but the no clear cut margin is given. I tell, like, ours was the new university, first pharmacy university. So the post of veterinarian, they did not even sanction. And we are telling that this is, you have to sanction this also. So such things were ignored. It was a bad thing. On one side, you are telling the pharmacologist, the department head, or the principal, why this is not there, why that is not there. He will say, sir, higher authorities are not allowing. We say that CPCA, according to this regulation, it has to be there. But then they will ignore. But if there is a clear-cut mandate that no, when government allocates funds, see, when it is a startup or there is a startup funds or like that, separate funds should have been given. Okay, now we are implementing this thing, so that separate funds should be given to the every college or university. When the computers came, they were given. So when the posts are there, yes, it should be with some norms. The veterinary post should be there in the gazette also, but it is not there. So these things have not been taken care of. But like even the facility of incineration, nobody is planning that. Still, many times we have to have the digging done and bury the animals. So, what I'm trying to say, on good thing has happened, yes, things are better. Animals are also, you will love to play with the animal, but at that time, but at the same time, they are now in a good condition. But still, something could have been done. Now, the things are that are animal studies justified at all level? I remember thousands of frogs and all, they used to be killed in the higher secondary. Although if the person is going in engineering, he has to kill the frog. And of course, the frog is totally gone. But many times it has happened that one thing which has occurred after this regulation is that, yes, unnecessary experiments have been totally removed. So good thing has occurred. There is a restriction on the number of animals. Bad thing, yes, dog experiment, which I still feel that it could have been done. Now, I mean, animal activists, they may not like the things, certain things, what I'm telling that like, yes, dog experiment used to be so good as a pharmacologist that we used to learn a lot of things. Dog experiments used to be of that type. And if instead of dog bites and all the cases have occurred and they are really worsening because these courts are giving directions of now. So that is also a job of that we should consider the animal welfare from that point of view. That there is to be controlled. Yes, the vasectomy and all those things have started, but still lot. But it is not something very good. And in India, totally they got out. I mean, people are going to neighboring countries to get their experiments done. So those things have been little bad. Monkey, for example, it happened, but somehow it was managed. Thanks to Dr. Balram Bhargav at that time, in the COVID time, he could manage and do it. There have been things. And the ugly thing sometimes I feel, who need the most, like, if somebody has to give the IV injection, it used to be a good practice from physiology experiments point of view, the pharmacology experiment for a doctor. Now they may be okay, they have the models and all that by which they can practice. But it was not a bad idea, but somehow it is gone. Another thing is the Schedule Y of toxicology, that you have to do acute toxicity, chronic toxicity, subacute toxicity. Are they required of every time? Without naming the institution, I will tell you, there was an institute, I was there. They used to, every time the animal was loaded with the toxicology experiment, I said, what for you are doing? Oh, the chemistry department, they want to test 10 compounds. So without doing toxicity, we should not proceed for the efficacy. I said, it is a foolishness. You should first prove efficacy, then go for toxicity. Same thing in herbal. If you have several extracts, why to do toxicity first? See that whether it is effective. Toxicity should be last. That okay, now drug is 100% it is going to be effective and useful. Then you go for toxicity. Then another thing, I asked once the DCGI people, you tell me in these 20 years, how many products you have rejected on the basis of just toxicology, or how many R&D, the work where it is going on, the drugs have been removed. Oh no, it happens. Then why the case of this nitrosamine has come? I mean, ranitidine, the cancerous substance was found. They said, oh, in the manufacturing process, there can be some adjuvant which has gone in. Well, it is correct. But how much justification? So that sometime it becomes a really ugly thing to discuss on that part. The drug has not come, their efficacy has not been proved, and you still want to do the subacute and acute toxicity. Or if it is done, there can be some limitation there wherever possible. If the drug is not going to be used in pregnant women, why you want to see the teratogenicity or like that? So those things slowly and slowly they are getting refined and revised. Now comes the replacement part. The good thing is that yes, the human techniques are taught. Computer simulation experiment, excellent, let me tell you. But yes, there are limitations. It is good if simultaneously, at least at PG. Now, simulation in PG is again a wrong thing. It should be their PG student of pharmacology at least has to do some experiment. That is what is still like. The PG, you cannot get away like that. And then, wherever possible, biochemical and biotechnology experiment have to be introduced. That is the most important part of it. Right? So, good thing has been there. Yes, because of this, these things like good come. Now AI, I don't know what AI will do further. But certainly biotechnological experiments, they are definitely better. The bad thing is, at least some training has to be there. And I remember in the Trends in Pharmacological Sciences, once the article was written by somebody that we don't find any in vivo pharmacologist. I mean, one who is trained for in vivo experiment is not possible. Same thing happened like in COVID when we developed one drug and we were saying that everything is because of the angiotensin converting enzyme 2. Somebody asked me that how will you explain the deaths occurred after the ACE inhibitors? I could explain. The reason I could explain was the thorough background of all pharmacology, the in vivo, in vitro, and all others, multiple, which is not possible. So unless you do in vivo, you cannot explain certain things. So that is there. So it cannot be totally zeroed down. Ugly, yes. I mean, the all members of these IAC may not be expert. So sometimes when they become rigid, no, you cannot do this, or you are expecting everything from the computer simulation, it is not possible. Like rabbit, I mean, for the pyrogen testing is very important. But lal test versus rabbit, both have limitations. At times, you may have to do with the rabbit test also. So that replacement, one should not be rigid. That's what I want to tell. But certainly, at times, good science is lost because of that. Well, refinement is definitely, it is there. One of the refinement, I will say that herbal extracts. Now, many times it is like a blind thing in herbals also. But when you know that phytochemicals, can you not imagine from the basis of phytochemistry what actions will be there? I'm talking about the structure activity relationship or quantitatively structure activity relationship. Whole world, I mean, if you see the 20th century, every disease has biomarker. every disease, we know that this is the receptor, or this is the protein, or it is the second messenger, or kinases, or phosphorylases, or mitochondrial thing. Most of the things are known. Why the resistance occur? Penicillin resistance because of penicillinase. Now, for that, you need not to do in vivo. Directly see the test and go further. So, what I'm trying to tell is that all these things have occurred only because of the restrictions which have occurred. The restrictions have got all this. So, it's a good thing which has occurred. Better planning, research methodology is now properly understood by every PG student. Previously, in our time, in 80s, we used to give the training, special training for that, but there was no separate paper for research methodology. Now we have everywhere, it is a research methodology. So this thing has occurred good, but again, the bad thing is that yes, many times, I mean, that too much delay even today on the large animal experiments is also not a good thing if it has to be outsourced. The ugly part, I will say, that now when we say oxidative stress or like that, now the various disorders, you need not to, you cannot unnecessarily, I mean, you have to reduce there. I mean, the refinement has to be done. If it is not possible, then it should be allowed. And sometimes we feel that those who are nominees of the government, they have to have more training programs for them also, so that the proper replacement can be done. Now, chick ileum instead of rat ileum, it's a good, but how many labs could do all throughout the country that? It was very good thing. Use chick ileum, that's it, for training purpose at least. But again, even, and what's wrong if the pharmacology experiments are done with the chick ileum? There the CPC is not required. So something could have been done there. Reduction, yes, this thing is definitely, it has occurred. The power of study, everything is now considered. And students are also learning what is the power of study. When they come to clinical, they know more, but now for human being, I mean, for the animal experiments also they know. So principles of humane are used. So very good thing has occurred like that. But unnecessary reduction is also not a correct thing. It has occurred at times. And research industry, they are also suffering because they have to do certain numbers. They have to follow the power of study. But what I feel is that, I mean, at least I had good experiences with every whosoever used to be the animal ethics member coming. I have been interacting and all that. So it's always they are agreeing, but unnecessary reduction is not allowed. But many times students take advantage of that. They also want to have shortcuts. That is not a good. Right. And the bad thing is that, yes, for herbal drugs, again, you may need it. And accordingly, things have to change. And again, you cannot reduce everything with rabbit test also there. Well, I mean, when you talk about the reduction particularly, 60 to 80% animals are used in the pharma are for the promising candidates, and they are the toxicity also. Reductions are possible. And large animals, dog, rabbit, primate, they also need to be there. And thanks that this 23 regulation which has come will pave a little better way, and these experiments will come into place. And again, before a human trial, certain things the DCGI also asks, CDESCO will also ask, the drug approval authority also asks. So that has to be redefined. There also, simultaneously, the changes should occur. and one should understand. So reduction will be more effective if this thing is there. And so animal reduction, refinement, you have to do that. Like they play very important role, test role, safety assessment and all that. So it is possible to reduce all these things by proper planning of the test. And in toxicology, as we told, there can we replace rodents with the other like chicks and other things, or can we take out from other like the zebra face has got and Drosophila, all that? Can we go and have certain replacements of that type? This is a question which can be continued. Now, again, the pain and sufferings. Yeah, very good thing is that even I used to stun the animals and then sacrifice. And I feel bad, used to feel bad at that time. Even those who used to see me doing that, they used to curse me rather. And now things are changing. It has been totally stopped. And but the bad thing is that yes, especially it comes to government colleges, that you need some support, financial support also for that to replace how you used to have there used to be chopper also coming in, or the more chemicals to be used. It can be done that way. But yes, things have occurred nicely also, but there are some limitations. Now, surgery. I would like to tell that for surgery, yes, there are recommendations for the anesthetic agent to be used. But how, I mean, if particular student is doing surgery, has he been trained for it? So that is another aspect which need to be done, and how surgical training are given. Slowly, if the animal experiments are not there, how they will be trained? And it cannot occur in the smaller rat or mice. This is possible, but at that time, those dog experiment used to be good, and we used to learn also. But so surgery is a little bit question, but one cannot be rigid in learning something. If goes, the member of ethics committee should allow certain things like that, and updation has to be done for everything. That's what is there. Euthanasia, very good thing that yes, it's a painless death most of the time. I will say, well, but certain experiments are only to see the stress, central nervous system stress. So there it has to occur. So sometimes those things remain, but anyway, things are going on to replace, refine, and reduce. So those things will continue. So having said that, question comes that, like in India, many times if we say that, okay, how many new drugs have you have come up? Does it mean that you are not using animals? Animals are still used, but the new drugs are not done. And in the countries where they have come, are they continuously killing mercilessly? Answer is strictly no. You are there the regulations came first. So these facts remain. A new drug discovery is again a task of several disciplines working cohesively, and that cohesiveness has to be think of. Now, previously, even the funds are also not easy to get for a scientist, and sometimes the shortcuts are used for the animal experiments also. That should not be there. Anyway, so I have just tried to tell that on one side, which is important to have the new drug discovery, you need animals, and you have to see that the drug discovery program does not suffer, and you have to be within the regulations and still keep on doing the experiments. Now, in my lifetime, I always used to think that will I bring any new drug? Yes, during COVID, I could do that. But yes, it was a dream only. And I used to believe that how this research, I mean, everything is a challenge. But what I learned from these almost half a century of life, I could that newer now the whole research has changed. Not that blind screening, I'm not talking that old things. The thing is that now you know clear targets available, biochemical or biotechnology based targets of every disease. And the new drug, when it comes, you have to pass through these two things first, which many times it is not done by even pharmacologists. Analysis, yes, better and better analytical methods are available. And yes, dosage forms, that is also a new drug. So things have changed a lot. And I will say that as a pharmacologist, I have seen again all these changes. My career started in 80s and in 40, 50 years. I mean, chemotherapeutic revolution was that like antibiotics when it was discovered, penicillin and biological revolution when we started realizing the receptor theories, molecular revolution when subcellular mechanisms came, and then human genome project changed everything. Based on that, the omics revolution had come, and now new revolution is the post-COVID era. Well, so one of the biggest change which I have seen in the drug discovery is that previously we used to see the drug first and then disease. Now the disease comes first, the present. That means you should understand the consequences of disease, or rather the origin of the disease. That what gene changes, or what protein changes, or what mechanisms changes, and because of that you get a disease. So target that. So targeted approach has come now. And if you see the whole discovery process has changed toward the biotechnology side now. Question comes, yes, as a drug discovery person, how strong we are in the biotechnology or crystallography and all that. How much emphasis is given. That in our time it was too less. Slowly it is emerging. Hope that change continues, but acceptance from the other students also are there. So today the drug discovery is phase 1, phase 2 is all right, but everything starts with the understanding the protein, genes, and then you go for in vivo experiments. Now, this is a crucial thing that as a welfare of animals, again, before coming to this, you have to have everything done on the other side. That's what is required. And one can see how things have changed. I mean, pharmacology talks more. This is a figure from the simple Dale also. Whole pharmacology approach has changed. People are nowadays talking all about the AI, as if AI is everything and it is something exciting. I tell you, in 2002 itself, we have used AI and we published these papers. ANN network at that time. You can see in 2007, those publications came. And we have done that. But AI also has limitations. So when we talk of like the simulation, there are limitations. AI also have some limitation. See, one of the things is that they have no intelligence. When in vivo experiment used to be there, we used to have certain things which are emerging. We used to see the dog and those animals, and we used to observe certain effects of biological effects, which are again otherwise not possible. In AI also, it is not intelligent. It is based on the data which has been fed to it. So machine learning rather. So, like, if I simply compare, it all depends on what is fed to the computer. That's how the AI will work. So just like that, as AI, even the simulation experiments, one has to have that limitation in the mind. Well, so ultimately the use, good chemistry, good target understanding, that is what is the key. Based on that, you can have high throughput screening or anything. And based on that only you can get new drug. So this is just a side tangent. So pharmacology has been there. So, with this, like, remember when I started, I was not knowing ABC of this computer. Imagine when all these things were there, no regulations, and we were doing, and we were trying to have a new drug discovery. I don't know. I was like a lost soul. Psychic. I was a really psychic person then. If I do the experiments, if I want to do a drug discovery process, that means I'm a psychologist. I'm a psychist. It happens. So, see, the whole thing previously, the revolution was like in vivo dog experiment. That was our era. We used to do all these things simultaneously. We got out of also many things. When receptor theories came, I remember in 1990 when I was there in the postdoc in the Canada, I was excited when I could learn polygraphs properly. I was excited when I got radio receptor assays done, then cloning also done. But by the time I learned this, things have changed. But I feel pride that when I used to do that simple experiment on rat, we used to contract, we used to have potassium chloride given and dose dependent relaxation by histamine. Rest all was all using different drugs and all that. That was a simple experiment. No hi-fi equipments, nothing. And this is my actual PhD thesis slide. And this was the outcome what I got. Just based on these simple experiments, this was the outcome. I could show how histamine, noradrenaline, alpha receptor, beta receptor, alpha receptor, alpha 2 receptor, how uptake is occurring. Even the biochemical tests were also less. But based on the potentiation and all interventions, I came out with such a hypothesis. What I'm trying to tell here, that when a PhD student used to come, and I always say even today, that I used to tell him, I never believed that, I believe that this person is all throughout night in the lab and that's why he's a good student. No. Apply your mind continuously. 24 hours, your mind should be continuing on your project, work. It doesn't mean that you have to be in the lab and doing the experiment unless. So, planned experiment, understanding the basis, understanding you. Do one experiment and have 10 interpretation. Do 1 hour experiment, spend 10 hours of thinking on it, what you got. That's what is the approach required. That is what is one message which I want to tell. Now, again, things have changed very fast. In the 80s and 90s, we used to learn simple structure activity relationship, and now it is a quantitative structure relationship, and now molecular modeling and whatnot. Similarly, biotechnology is the exception. But the question is that even today, if you take all drugs together, around 40% drugs, they work on enzymes. Around 30% drugs work on the G protein coupled receptors. And rest come the last. Now, if you see the amount of research going on toward these 7, 4, and all this small percentage is very high. And we don't concentrate on this. This is a question to be kept in. So through Human Genome Project, we got several genes, several targets, but drugs, success of the drugs are again less. Well, forget that. And now, especially after COVID, all the more there, but again, for last 2-3 decades, must have come from the like Ayurveda or Unani medicine, Siddha medicines, or traditional medicines, what we say, or herbal drugs. And people think we should have our own traditions. Well, we have got Ayush department, so why not herbal based drugs? So global thinking, but local actions. That used to. So in that process, reverse pharmacology came in. Reverse pharmacology means you start with the activity, you need not to do old theory. You understand the receptor and all that, and based on that, try to get hits, lead compounds, and then get the final drug candidate. That was given by Dr. Ashok Vaidya and Dr. Ale Bashelkar and our Bhushan Patwardhan, Dr. Bhushan Patwardhan. They were the real pioneers in this. And they showed the successes of various drugs. I mean, you reduce the number of experiments required, time, duration, money, everything is same. Well, we also had the similar approach. One of my approach was for the plant Anicostema latrode. Now, this we selected out of the market survey. It was not that I used to be a classical pharmacologist, never the herbal, but somehow I was labeled as the herbal person. But I'm not feeling bad. But what I'm saying is that the approach was little different. I used sort of a reverse pharmacology approach, only knowingly or unknowingly, not knowing the concept. They published later on, Dr. Ashok Vaid and all, they published the papers on the reverse pharmacology, but we really did it. And we started with the animal, instead of animal, the experiments on using the patients only. And we could get that. From the patient study itself, we could get that a drug is acting not on glucose directly, but also on the lipid. We could show that, and even the genoprotective effect we could show at that time. So effects it is there, it is going to be permanent. So, why I'm showing is that it is not necessary always that good research will come out with the animal experiments only. Proper planning, more time devoted to the thinking, outcome, and what we are expecting, and the planning properly, it will give the results. One more example I will give that like COVID. COVID was havoc. And previously we used to say new drug discovery means 10-year time, 15-year time. And now it is known that, and in COVID, we wanted within 6 months the new drug formulation. And a lot of things happened at that. Drug repurposing is one of the concepts which has emerged in that, wherein you need not to do too many toxicity study and you get better results. Drug repurposing. And I at that time has initiated this. We have thought about it. It's a reverse engineering, I'm calling this. I'm just trying to give various alternatives. There are experts who will tell alternate animals to be used, but I'm giving alternate method of getting a new drug. Now, reverse engineering in the engineering, like you take a machine, dismantle it, try to find out what are the problems, and reassemble with the improvised. So what I did in phytochemistry, or in a herbal based drug, you need not to isolate compound again and again. See the literature, you will know certain compounds, and based on that compound, do simple docking studies with the computer itself. Because docking with what? Docking with receptor, docking with protein, docking with particular disease biomarker. There you have to be selective. You have to have a thorough understanding. In COVID, when we found ACE2 was there, angiotensin converting enzyme 2, we came to know that 81 receptor, 82 receptor, RDRF, which is multiplying the virus, and ORF, because of which there was the death of the cardio. We took all those 5 targets on one side. Other side, we did the compound, the herbal, some Ayurved person, some experts of the ethnobotany, they told us that these are the likely compound they may be effective, likely plants. We did not go into extractions and all that. We just tried to have the phytochemicals in all. And then based on that, we made the formulation. So, and we were successful. That's why I'm showing that reverse engineering can be done. And based on that reverse engineering, I mean, as I told you, phytoconstituent, disease molecular specific targets, do that. And for dismantling means phytochemistry, you should know, the chemicals which are there. And analyze whether they are effective, the disease targets. We did that, SARS-CoV, we identified targets. In those days, you can see the September 2020, we published that. And based on that, we identified the results. And finally, out of various hits, 3 lakhs hits compounds, the docking study compound, we could get 26 hits and then 10 lead compounds, which were effective on ACE2 and all that. And when we made that formulation, it was effective. Not just theoretically, we got the study done in the University of Missouri and we got the results. And even the clinical studies proved it. So what I'm trying to say that this can be another approach to reduce the number of animals and better outcome. So this is the way it can be done for other diseases also. You first look at other aspects rather than just this Ayurveda literature says this, so let us try in this patient or in this animal model or like that. Based on that, you go back to your chemistry, because pharmacy is a discipline who is expert in chemistry as well as formulation, as well as pharmacology, as well as cognos. No other discipline is like that. So utilize that, and based on that, try to develop the formulations. I am sure you will get a good success. So thorough chemistry, thorough analysis, thorough pharmacology, and you will get the good formulation. So we had all these things, considered it, serendipity also based on what has been told and all that one can do. And be choosy. Like analgesic. Do you really need a new drug for analgesic if it is like aspirin and wonderful drugs are available, or any local anesthetic directly? So there are many other important, like microbial is a very problematic, even today, resistance. And plants can give a better answer there. But the work is not done there. Everybody is after this anti-diabetic and anti-hypertensive. That is also another thing. So let us be little careful in thinking. So what I'm trying to tell is that to have a better outcome, this is the way that you change the approach for the drug discovery. That is what I'm trying to emphasize, consider. And toxicity, as I told, has to be done once everything is clear, rather than doing toxicity for everything, and then you know that this compound is not effective. What is the use of that? So this should be the approach. And yes, I can simply say that I mean, some suggestions for those who are, I mean, like the clinical, I mean, I will say to the CPCA members also, be reasonable, but at the same time, try, it is our duty also to be reasonable in presenting particular protocol. The processes of approval, they should be done. And again, a good thinking on the large animals also, that is also required. Right. So this is what I have tried to sum up. I have not touched upon the alternative methods of the like geographies and all that. But they are, yes, there are different behaviors, but many times human simulation experiments become important. So there you have to see how can we do that. So I have not touched upon more in that, but all the methods in my life, I did not use in my career these new animals. So I don't want. But yes, a drug discovery, I can claim that yes, I could get also. So I have tried to cover all across what I mean from the point of view of animal welfare and use of animals and all that. And I will be happy if the questions are there and we can have more discussions rather than that. Or if you have anything particular topic on which I have to throw line without slides also, I can do that. So I will stop rather. I mean, I thought, I mean, more than 1 hour to, I was given around 90 minutes, but I thought let me have more discussion on that, if you permit. But certainly, if you want me to touch upon particular, it will not be difficult for me to immediately reconcile it. So with this, I stop right there sharing the slides and I will be listening to the questions there.
Thank you, sir, for your valuable and insightful talk. Now the platform is open for the discussion. If the participant has any doubt or query, please feel free to ask, or you can post your queries into the chat box. If anybody having the question, please ask the questions, or you can post your query into the chat box.
Anybody from the CPCA nominees or those who are going to be speakers also, and I will be happy to listen to them. It seems that I have finished little earlier. I started little early, but I never thought that it will be.
Yes, sir. I think there are no more questions for the participant. If
So-
there are no further questions, I would like to conclude the sessions. Respected sirs, on behalf of organizing committee and all the participants, I sincerely thank you for your enlightening and stimulating session on medical experiment on animals. Your explanations of how to balance the scientific progress with ethical responsibility.
There is a question.
Yes.
Yeah, from Bhagyashri. What is the future? Can you repeat that question? In the chat box, it has come. Okay, what is the future considerations for animal experiments according to CCSEA? Of course, it would have been better if the CCA person had. See, animal experiments, they will continue to be there. I mean, you cannot get away from that. Only thing in the curriculum, in the our B Farm program or like that, it has been stopped. It has been reduced to almost minimum. I will say in medical college, it is almost very less. It is almost not there. In pharmacy, it is still continued thanks to our all leaders who have maintained that no, it cannot be, you cannot go away from that. But when it comes to animal experiment, previously, like we used to do dog experiments for physiology, which has changed. I will say, see, for dog experiment, sorry, for the frog experiment, we used to show the effect of fatigue. There is a contracture coming up. Now, physiotherapists, they found that myofascial trigger point, a sort of a knot is found after repeated stimulation of particular muscle, which may occur because of the activity also. And we used succinylcholine to damp that, and without any animal experiment, because it was the ointment which was prepared, and for that you were not to have that experiment. So, like that, what animal experiments, they are reduced. The future is that yes, it will be reduced, it will remain reduced, but for certain, like there was one conference in last month, only in February, and somebody had come from Hungary, and he showed dog experiments on arrhythmias and fatigue. It is possible. So it is something like we are going to be running away from the animal experiment. Do we really want to run away, or we want to do something? That is what is important here. So future, I can say, I mean, if you are asking the animal welfare people or those who are in the government, they want to control that 3 R's. Number one, proper facilities, proper food and all, water, and humane approach. That will remain. So their job is not to stop you from doing research. Their job is to facilitate the research. So in future also they will be considering like that, unless you feel something else. So the one who has asked the question, do you have anything in mind more than that? Can you just open up your, please? You
No,
can
sir.
answer. Yes.
No. Yeah.
Yes, Bhagesh, yes.
Yes, yes. Sir, actually, I also want to ask you that in vitro experiments are now replacing in vivo. So, what will be the effect of that now in the research area of pharmacology?
Yes, that's what like we started realizing in around 1998, 90, that nobody is available to do the in vivo experiments. They are, because not a single training is given to them. So if that thing goes away, it is going to be a real problem, because how many will be knowing how to dissect dog or how to dissect particular things? See, human being, no, every time you cannot bring a doctor, MD, to do that experiment. Even ECG is, or the in the rat, ECG is done, or echocardiography is done. Now, echocardiography in human also it is done, but from there they could come down to that. But initially, they were the cardiac surgeons only who used to come and do the experiment in the lab. So ideally, it should be a mutual process. Many times what happens, the people feel good that, okay, animal experiments are not to be done, so no animal house, nothing. So managements are also very happy that, okay, no more expenditure of that. But as a pharmacy, as a pharmacologist, we have to be concerned in that. And that is the reason that in pharmacy curriculum, still there is a good amount of animal experiment. There are some demonstrations also, and many colleges are still following it up. So it is up to us, our own interest. And I'm happy that, yes, you have shown the concern that if one should raise the voice that if it happens, like the CCSEA rules have been implemented recently, maybe because of that large animal experiments only. Because when the COVID vaccine was to be developed, they had tough time in doing that. So they are trying to revise it. So by expressing your concern slowly and slowly, it will be realized. But yes, once for all, the planning, everybody has started in a nice planning way, in the planned way to do any experiment. So it's a good thing.
Thank you, sir.
Yeah.
Excuse me, sir.
Any other question, any other thing, any comment also?
Yes, yes. Sir,
Yes, Dr. Shankar.
Yes, can you clarify this minimum number of animals which is required for the in vivo?
No, voice is not clear. Can you
Minimum,
type or
what will be the
or you
minimum
can
number of animals required for the animal experiment for in vivo?
small animals? No.
Small and
Can
large
somebody
animal for the statistical analysis.
can somebody explain what? Can you
Sir,
type out
Sir, he is asking how many animals are required for the statistical animals, statistical significance.
for
test.
Yes, sir.
statistics?
Minimum
Okay,
animals,
okay.
large and small.
See, in our time, we used to use only 3 animals and we used to get good results also at that time. But now, the power of study based on that p value and the expected results, we can calculate. Like for the clinical trial, what we are doing. And many times when we are reviewing the paper, that n is equal to 3 or 6, they become they are acceptable even today throughout the world. Right.
Thank you,
So,
sir. Thank you, sir.
like in the schedule Y of the CDSCO, they mentioned around 10 or what number is required. So you have to follow the regulations there.
Yes, again, anybody want to ask the questions? You can ask the question directly or you can post your question in the chat box. Sir, another questions are there? No. Okay. If there are no further questions, I would like to conclude the sessions. Respected sirs, on the behalf of organizing committee and all the participants, I sincerely thank you for your enlightening and stimulating sessions on medical experiments on animals. Your explanation of how to balance the scientific progress with ethical responsibility has helped us better understand the importance of human research practices. The way you clearly discusses the current challenges and new advancement has motivated us to conduct the research with greater care and accountability. We truly benefited from your vast experience and valuable guidance. Thank you, sir, for your precious time and for sharing your knowledge with us. Now the next session will be started at 1:30. I request all the participants please join sharply at 1:30 by using the same link. I request all the participants to please switch on your camera to take the photograph. I request all the participants to please switch on your camera to take the photograph. It's my humble request to all the participants. Thank you, sir, for your valuable sessions. The first session
Thank
is
you.
okay. Thank you. Now the first session is over here. The next session will be started at 1:30. So I request all the participants sharply joined at 1:30 by using the same link. Thank you.
Okay.